session is by Dr. Keshav K. Singh. Dr. Keshav K. Singh is a member of Center for Free Radical Biology and Center for Aging at University of Alabama at Birmingham. He also founded the Mitochondrial Research and Medicine Society in USA. I would like to invite Professor Dr. Keshav K. Singh for the talk.
Well, thank you very much. What I'd like to do is to give you a different flavor to what you've been hearing this morning, and that is to do with mitochondria and mitochondrial genome and how it relates to cancer health and cancer disparities. So basically what I'm going to be doing is talking to you about how mitochondria ancestry affects the cancer health and relates to cancer disparity in very different populations. One thing I do want to ask you to please not record the presentation because I have some data which I am sharing and something I do not want to spread. These are my disclosures. I founded UA Biosciences. Allergan Aesthetics. I've been involved in the Faculty of Aging and Life Norm and beyond medicine and breast cancer young women. What I want to do is, because it's different and many of us don't think about mitochondria, mostly we think about the nuclear genome and its role in cancer. My job is to take you to mitochondrial genome and how it relates to cancer. So I'm going to give you the I'm going to give you the basics of the mitochondrial biology and genetics, talk about mitochondrial role in pathology, and then talk about the role of mitochondria in cancer. Basically, I'm going to give you the mitochondrial yin and yang. So we know that the mitochondrial genome is circular in nature. It interacts with the nuclear genome. So obviously, it's a one cell, two genome story. And when we think about the mitochondria, there are hundreds of these genes, close to about actually 1,500 genes which make up the mitochondrial oxidative phosphorylation. So what you're looking at here is the nuclear genes which encodes the different mitochondrial subunits which will involve in the oxophosphorylation, electron carriers, the mitochondrial DNA homeostasis, and so forth. And then you find that there's secondary effect of the mitochondrial functions as well. And when we talk about mitochondria, we know that most of the time it's called the powerhouse of the cell, but mitochondria contains the several thousands of these proteins which are part of the many different pathways. What you're looking at here is the proteins which are localized to mitochondria, involved in, say, for example, iron metabolism, lipid metabolism. They're involved in the mitochondrial fission and fusion, signaling, and so forth. So it's basically to show you that the proteins which are involved in the mitochondria are involved in the different pathways, and therefore it's beyond the ATP production of the cell. Here, in the context of the oxidative phosphorylation, what I'm showing you is that the nuclear genome encodes, say, for example, here in case of the complex 1, 35 subunits, and the mitochondrial DNA is encoded by 7 subunits, and together they make a complex 1. So what happens is 35 subunits are synthesized in the cytoplasm, they are transported into the mitochondria, then they get together with the 7 subunit, and there are many different assembly factors. So these guys together, for this to make a complex 1, assembly factors come into play and they make the complex 1. Complex 2 is exclusively nuclear. Complex 3, the same story. You find that there are 10 genes which are encoded by the nuclear DNA. One is nuclear mitochondrial DNA, and so forth. And what happens that we think that in the context of respiration or mitochondrial ATP production, that these things are linear. So for example, what you're looking at here is complex 1, 2, 3, 4, but in fact what happens, they constitute super complexes. So for example, complex 1 can get together with complex 3 and complex 4, and that can carry out different function altogether. And same thing, there are a lot of permutations going on in the context of the the biology. But if you look at the context of mitochondrial and nuclear crosstalk, this is a classic example where 35 subunits are encoded by the nuclear DNA, synthesized in the cytoplasm, transported to mitochondria, and then get synthesized. And you can imagine that the retrograde regulation, which is part of the process in any human cell, and particularly very important in cancer, that's something we ought to pay attention to. So what I'm showing you is that the mitochondrial DNA encodes the 16 kb circular DNA. In some cases, it can also be linear. It encodes 13 proteins, which are part of the oxidative phosphorylation, and the number of mitochondria in the mitochondrial DNA varies from tissue to tissue. So, and also what we cannot ignore that the mitochondrial DNA encodes microRNA as well, and in fact, mitochondria functions as a reservoir for microRNA, which are encoded by the nuclear genome. And why they reside in the mitochondria is anybody's guess at this time. Then you also find there are hundreds of these peptides. So we think that, well, there are 13 protein part of the oxidative phosphorylation, they make the ATP, but no, there are many peptides, small peptides, and many of them are functional. So you think about a whole machinery in the mitochondria which involve 13 proteins, the oxidative phosphorylation, related microRNA, and then you find these peptides as well. And then if that's not enough, one has to think about the heteroplasmy of the mitochondrial DNA. So in normal human being, there is a different forms or variants of the mitochondrial DNA, which are described as the heterogeneity of the mitochondrial DNA or heteroplasmy. So and this is what helps us adapt. If you're living in Siberia, you don't want to produce ATP, you want to produce heat. So the mitochondrial function will change depending on where we are in what part of the world. But this is also creates a complexity in terms of understanding the cancer or any other disease for that reason. And not all mitochondria are created equal. So depending on the cellular demands, mitochondria can be different cell type, exhibit variation in molecular composition, morphology, and function. And here is an example where you find their cell type is specific profiling of brain mitochondria, where you find the mitochondria proteome in astrocyte to other cell type differs as well. So there's a lot of variation. It's like a population genetics within the cell. And that mitochondria talk to each other. And if you look at this particular slide here, they are dynamic organelle, and this is constant fission and fusion going on, and within milliseconds you find this fission and fusion obviously changes depending on the physiological state of the cell or the diet or what we're eating and what we're drinking. And here you see that there are examples. For example, let me get back here. This particular mitochondrion fuses with others and get together again. And this is particularly one cell. Imagine there are trillions of mitochondria in our body and what they are doing and how they interact with each other. Then if we talk about there are mitochondria conversation within the cell. So here's something we are looking at in the context of cancer, because in microenvironment it's very important that how mitochondria can transfer from one cell to other, or the tumor or non-tumor cells. So here the mitochondria interaction you're seeing is with the other organelles. So constant this fission and fusion, not only between the mitochondria, but they interact with endoplasmic reticulum, they interact with lysosomes, and so forth. And then there's a conversation between the cells as well. So here's an example where you see these are green mitochondria, and what you see here that from one cell to other, these green mitochondria are transferred and we do not know whether it's a bidirectional or unidirectional. So something we are in the laboratory trying to tease out in the context of microenvironment and tumor. And then this is one of the fascinating one. What you find that the migrating cell leaves behind a trail of mitochondria. So imagine if you are a metastatic tumor cell, how are you going to migrate, how mitochondria are involved, is anybody guess at this time as well. Excuse me. And can I just one second? This is water. And this is one of my favorite slides where I see that in the normal human cell, there are cell-free mitochondria in the blood of normal human being and in ICU as well. So what you're looking at here is there are close to about 4 million mitochondria floating around in our blood. And we have no idea how this plays a role in the context of cancer or any other disease as well, and whether these are involved in tumorigenesis or any other pathways, something we are trying to figure out in the lab. So what about the role of mitochondria in human pathology? So in the context of mitochondria, there are two sets of diseases, mitochondrial disorders, which are primary in nature, where mitochondria is the primary cause of the disease. And what you're looking at, the inner circle, which are darker green, are the mitochondrial diseases. On the outer side, you're looking at the secondary diseases where mitochondria are involved, including aging. The mitochondrial diseases, something we have been working on for a long time to make headway in terms of defining it, what mitochondrial diseases are, how rare they are, how common they are, something I started a foundation and the society in US and in India as well. If you're interested, you go to mitoindia.org. But the problem is that because it's a multisystem disorder, the definition was that if the 3 organs are affected, suspect mitochondrial disease. Because it's a multisystem disorder, it takes on an average of 14 doctors to diagnose a mitochondrial patient. This is when you are rich and famous families, which I have helped get diagnosis. It turns out that, for example, if you look at that meninges, so meninges is myo, neuro, gastrointestinal encephalopathy. There are several organs that are affected, and you go to an expert, and that expert, say a nephrologist or somebody else, neurologist, and they will look for the neurological problem, but nobody connects the dot. There's something we have made quite significant progress in US, and we continue to make in India as well. On an average, 1 in 2,500 children born in US are affected by mitochondrial disease. And because it's maternally inherited, it's quite significant in population where you find consanguineous marriages. What about the mitochondrial function declines with age? We know we have low energy. And something during the COVID, we got involved where we had certain idea that how COVID may be involved in pathogenesis of the COVID disease. What we showed first was in August, actually the COVID started in 2020 March. In August, we had an article showing that how hijacking of mitochondria, of host mitochondria by COVID-2 may be involved, and why the elderly were dying much faster than the rest, or the people who had diabetes and other things. Then this was a collaboration through LinkedIn. I have not met the group. I collaborated with England in King's College London, where she read the article and said, well, can we get together and look at the patients indirectly? And turns out that what we showed was in fact true for the patient as well. So there's a mitochondrial metabolic manipulation for SARS-CoV-2. This became a pretty interesting article for many others. So there are 30,000 downloads some couple of months ago, the 21 downloads, and we got the attention of the White House to talk to Dr. Fauci and Dr. Birch about what can be done at the time of freeing the society from COVID. So what we found is quite significant, that when we looked at the mitochondrial release of the cytokine, which we call mitokine, and that fibroblast growth factor 21 correlated very significantly with the severity of the disease. So what you're looking at, the healthy mitochondria in green, the mild COVID, and then you see the severe COVID, and the fibroblast growth factor 21 correlated very significantly. That's something we continue to work on it. So the bottom line was that if you have a good mitochondria, you have low risk. If you have a medium mitochondrial function compromise, you have medium risk. But if your mitochondria is compromised either because of the aging or certain diseases, there's a high risk of that. And this turns out to be very critical for what you find in the long COVID. So there are 56% of the people who have chronic fatigue, and that seemed to be the compromised mitochondria. So what about the role of mitochondria in cancer health disparities? As I mentioned, that mitochondrial DNA is inherited maternally, the sperm mitochondria is degraded, and that relates to very nicely for us that we can follow the human migration, every population around the globe, and look for their, what that relates to the mitochondrial function as well as the risk for cancer. And we have focused so far in Hispanic as well as the African population. And what is important to realize that this is what you see here, that from L1, L2, L3 from Africa, we migrated to Europe, and Thangara's group actually in India very nicely showed that there was a group that migrated to Andaman Nicobar, and they moved from south onward to the north, something you will not see in many other slides. But the turnout that there are haplogroups and then the haplotype. And we have, and others have shown, that we can take your mitochondria, we can put in the cell line, remove the mitochondrial DNA altogether, and ask how your mitochondria measures up with compared to the other people's mitochondria. So we have tested European, we have, in the European we got the German, the Scandinavian, and so forth, including the African population. And what I'm going to show you is the data, and I'm not going to bore you with all the mechanistic side. We have looked into it. So it turns out that NCI invited me to basically seed the idea to the epidemiologists who were looking for GWAS in correlation with the cancer risk. And turning out that many of those GWAS studies were not panning out. So my goal was to get them thinking about the mitochondrial DNA as well as the mitochondrial nuclear and crosstalk. During that workshop, I organized what we came up with, that there are many of these variants present in different populations. The one we have focused on the G10398, which is, I call it good to awful. And this is prevalent in African population, which leads to very high risk of breast cancer in young women. So what we thought, well, we can do is find those women and isolate the mitochondria from the platelet because they do not have the nucleus. So we will be transferring only the mitochondrial DNA. And we did that. We created a cell line which is devoid of the mitochondrial DNA. So you see in the red there. And then what we could do is we took the platelet, isolated the mitochondria, and transferred into the cell line. And turns out that we can recapitulate the tumorigenic phenotype. And I'm going to show you some of the very simple data slides. You can look at the paper that's published. One thing we looked at first was the increased reactive oxygen species. And turns out that there is an increased oxygen species in this population. You look at the reduced membrane potential in the G2A population, and then you find that they were resistant to apoptosis as well. And finally, we looked at the AKT, which is part of the survival pathway. That was phosphorylated, and AKT was playing critical role in resistance to apoptosis. Then we looked at the OXPHOS secretion. What turns out to be, we will think that there's a variant, and this variant will affect mitochondrial function, and often the notion is, well, it will reduce mitochondrial function. No, it turns out that this enhances mitochondrial function. The complex 1 is particularly up in regulation, and that seems to lead to the metastasis of the disease. So the increased oxidative phosphorylation, and now it's pretty clear in many of the studies which come out recently that increased oxidative phosphorylation relates with the metastasis of disease. Very quickly, do I have any minute or two? 2 minutes, okay. So we also looked at the mitochondrial DNA content in African population, and what we found that the mitochondrial DNA content was low in African population, particularly in prostate, whether we take the normal or the tumor. So we wanted to know what is the cause of the reduced mitochondrial DNA. So we had this study a long time ago we did in East. We mapped out. Okay. So we mapped out all the genes which are involved in the maintenance of the mitochondrial DNA using that East model. Let me get back to that. And we identified close to about 100 genes. We focused on one of the gene which is involved in the maintenance of the mitochondrial DNA replication and mitochondrial content, and that was the polymerase gamma. So polymerase gamma is made up of Pol G and Pol G2, which makes the whole complex, and that derives the mitochondrial DNA biosynthesis. So what we have looked in, and others have shown that the African variants in the gene impact the mitochondrial DNA stability an oxidative phosphorylation. And I'm going to show you very quickly that when we looked at the Pol-G, it made up of the three subdomain, exonuclease domain, the linker domain, and the polymerase domain. And we look for the variation in what we find in the African population. They were in all domains. So you see the exonuclease domain, we find the linker domain and the polymerase domain. And I'm going to show you the functional analysis we have done. So when we look at the exonuclease domain, for example, and we take the same approach we have done before, we create the site-directed mutation and look for D136, for example. When we express this, we see the heterogeneity of the mitochondrial DNA population, what we see in African, and it also leads to the effect on the oxidative phosphorylation as well. And at the end, what you're seeing is the complex 1 activity. Same thing is true for other domains as well. I'm not going to go in detail in the interest of time. But turning out that all these variants which we find in African population, they are functionally relevant and they lead to the tumorigenesis. Here is an example of that, where we genograph this in the mice and see the large tumors are formed, and these are African variants. And one should worry about in the context of clinical diagnosis, to treatment, and everything else. So in conclusion, what I've shown you is that African ancestry, particularly the good to awful 10398, induces aggressive breast cancer in mice. We can recapitulate that. And also in the nuclear genome, which maintains the mitochondrial DNA, also affects the mitochondrial function and is involved in aggressive cancer phenotype. One thing I also want to bring attention to, that I founded this Cancer Health Disparities Journal. So those of you who are interested, please feel free to communicate. And then if you want to know more about mitochondria, I can tell you that I'm a mitochondriac. So everything mitochondrial here. So for example, you can follow on the Twitter. There's a mitochondriac group on LinkedIn. There are a couple thousand people there. And then mitoscientists. And I have people around the world who actually contribute every week, bringing new articles and type which are relevant to mitochondrial diseases and cancer. Want to acknowledge the people in the lab. Trevor, Bhupender, Prachi, Noha, and Ved were the key people who contributed to this project, and I'll stop here.